Hello everyone and welcome to today's live broadcast, Interpretation of Hematology Veterinary Scattergrams in Normal and Pathological Conditions, presented by Dr. Kay Criswell, founder and owner and independent consultant at Westbrook Biomarker and Pharma Consulting. I'm Susie Valdez of LabRoots and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Sysmex. For more information about our sponsor, go to www.sysmex.com/US. If you're looking for multi-species hematology analyzers, go to the Products and Services menu button, click on Hematology Multi-Species. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located in the far left of your screen. Type your questions into the drop-down box that appears on that screen, and we'll answer as many questions as we have time for at the end of the presentation. If you're having trouble seeing or hearing this presentation, click on the Help Desk button located at the promotional board at the bottom center of your screen, or use that Ask a Question button and let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the accreditation button located in the promotional board at the bottom center of your screen and follow the process for obtaining your credits. Please join me now in welcoming our presenter, Dr. Kay Criswell. I now will turn the presentation over to her. Welcome, Dr. Criswell.
Thank you. Today's presentation will focus on the interpretation of hematology veterinary scattergrams. As a disclosure, I have received an honorarium from Sysmex for today's presentation. And importantly, we will be looking at a multitude of case studies. And it's very important to recognize that not all case studies are identical. And although we can use them as a good learning opportunity, one case does not necessarily pick next. The scattergrams that we're going to look at today were predominantly gathered from Sysmex analyzers, and in particularly the XT2000I and the XNV, both of them hematology analyzers specified for animal use. The Sysmex XN platform actually can be used for both human and veterinary applications. The XN is a product of growth from the more original series, which was called the Sysmex XE series, but the XNs for humans has actually been in use since 2012. There have been multiple updates of footprints and other efficiency and scientific improvements since then. When we look at the veterinary applications, the first application for Sysmex was the XT application, and it had a release date of 2005. So it has a good history to it with over a decade of being able to analyze hematology samples with this technology. The conversion to the XNV or veterinary application is relatively new with this past July. Therefore, you're going to find in this presentation, I've actually put a couple of human applications. We call them one of the other multi-species, and you'll find that it's very relevant since this is a new release for the XNV. Using some of the human application will show you what you might expect to see in an animal population. So both XT and XN platforms utilize laser flow cytometry for cell characterization. As the image shows, the analyzer uses a laser beam at 633 nanometers, and as that cell passes through the light, it has the ability to scatter light, and the analyzer has the ability to capture that light scatter. The light scattered in a forward direction will indicate cell size. The light scattered in side scatter will determine internal complexity, such as the lobularity or lack thereof of the nucleus. And lastly, the Sysmex technology uses an RNA-DNA probe so that we can also capture fluorescent light. One of the first channels we'll look at is called the WDF channel. and this is the channel used for the predominant WBC differential. The scattergram on the left shows you the populations that normally can be discerned when running a hematology sample through this particular analyzer. We see several categories, the pink being typically our lymph category, the green in this particular case being monos, the aqua being a combination of neutrophils and basophils, and the red being an eosinophil population. This information is captured by capturing fluorescence, which is our DNA marker on the y-axis, and side scatter light, looking at the complexity of the cell. So it makes sense that cells that are more complex, the ones who have the biggest granules, the ones that have maybe the most lobes and nucleus, should be the farthest to the right. And those cells that are the largest in size should be higher on the y-axis. From this, we can see this particular channel allows us to differentiate lymphs, monocytes, neutrophils, and eosinophils. But we will see as we look at case studies, this channel is also going to allow us to predict that there may be atypical lymphocytes or immature WBCs. So to summarize this WDF panel, or the white cell differential fluorescence panel, we find that the leukocytes are classified according to content of nucleic acid, be that RNA or DNA, and the internal complexity or side scatter of light. In healthy individuals or animals, we find that there are 4 different populations that are clearly discernible. We'll find that our baso numbers actually need to be determined in a different channel. So in essence, our immature granulocytes can give us a 6-part differential when we see that we can actually also determine immature granulocytes. A difference in the XN technology is change in algorithms. And this change in algorithms, abbreviated SAFLAS, or Seismic Adaptive Flagging Algorithm Shape Recognition, actually incorporates a linear discriminations of cell clusters, allowing a better differentiation between the 4 normal populations in the WBC differential. All right, so that other channel, that WNR channel, the WNR channel stands for white cell NRBC scattergram. This particular scattergram, although it lumps together all of our other white cells that we talked about, it has the ability, using forward scatter and side fluorescence on Y and X axis respectively, to be able to differentiate our basos, since they will be above our normal population, and it will also differentiate nucleated red blood cells. So NMR provides identification of basophils, nucleated red, and we also have a debris area, and most people discount the debris area, but we're going to find in these case studies that even the debris channel can be important in interpreting abnormal scattergrams. Examples that you might find in that debris category might indicate there's a platelet clumping problem or potentially even parasitic infections. So a couple of other scattergrams that Sysmex provides for us is a reticulocyte scattergram. Reticulocytes, we recognize, are red blood cells that have been released from the bone marrow and still retain RNA, but no longer retain a nucleus. As such, we can stain that RNA, and we can, as shown on the image on the right, be able to determine populations of reticulocytes that have low fluorescence, LFR, moderate fluorescence, MFR, or high fluorescence, HFR, high fluorescence reticulocytes. This indicates the amount of RNA remaining in the cell. It actually is a good indicator of bone marrow activity. The more RNA present, the closer to the time of release from bone marrow. So this immature reticulocyte fraction becomes an important indicator of bone marrow activity. Both the XT and the XN platforms are going to give us reticulocyte counts, give us in both the absolute and percentage manner, and also this immature reticulocyte counts. And we do it, as we say, by having the y-axis being forward scatter with size. Our red blood cells are much larger than platelets, so typically, particularly in normal samples, we have a good separation between red cells that are much larger than platelets. Another scattergram that is present on both the XT and XN platforms is called the Platelet-O Scattergram. Platelet counts can be obtained on either analyzers by two methods. One is an impedance method, simply counting through the age-old method of breaking in an electrical current as the cell passes through. But optical methods can incorporate the use, in this particular case, of a fluorescent dye. And we're going to find that it allows us to have greater separation of red cells and platelets, particularly when there are pathologic conditions, such as microcytosis, schistocytes, or other red cell fragments that could be the size of platelets and be erroneously counted as such. Or in contrast, giant platelets, which can be the size of red cells and be counted as such. So the XN brings us yet another platelet scattergram, and this platelet scattergram is now called our Platelet-F Scattergram. And in it, Sysmex has incorporated yet a different platelet-specific fluorescent dye, which in this case will stain rough endoplasmic reticulum and mitochondria. With this particular scattergram, we are looking again at forward scatter, so red cells are large, platelets are small. But in this case, where our x-axis now shows us side scatter fluorescence. And by this type of algorithm, this type of separation, we find that we get even better separation between red cells and platelets under those pathologic conditions. And we will see a few case studies as we progress. So let's start looking at the veterinary scattergrams. This is our first one. I put this as a representative scattergram. This is a dog. And we will find from the ones that I've shown you already that perhaps it looks a little more linear. It means that neutrophils and lymphocytes and monocytes are kind of more linear. That's because although morphology is similar between species for white cells, it is not identical. And there can be various differences. There can be differences in lobularity, there can be differences in granularity, and that's what's going to cause the cluster formations of these various cell types. So it becomes extremely important that we're dealing with species-specific software when we try to do WBC differentials because of these subtle differences, but yet important differences between our white blood cells. We can find, however, that once we learn how to interpret abnormalities in these scattergrams, that they're going to be very helpful for you trying to predict pathologic conditions, even across species, and even though the basic scattergram may be slightly different. And you're also going to find that if you are familiar with former versions, such as the XE or XT, that if you convert to the XN, you will find that those pathologic scattergrams, again, are going to be highly similar. So let's show you a publication from ASCVP in 2012. This was represented as a typical presentation of dog scattergrams collected on the Sysmex XT. Here's that WDF scattergram we talked about, and we can see we have a good neutrophil population, lymphocyte, mono, and EO population. When we look at the WNR population, again, we can see the basos pulled out readily. When we look at the reticulocytes of this dog, we find good separation between red cells and the much smaller platelets. We see very few reticulocytes in this dog, but that still could be normal. Here is our Platelet-O that we talked about. And again, we see good separation between red cells and platelets. And again, we can now see immature fractions for both reticulocytes and platelets and measured as we need. And the last two scattergrams that we haven't talked about are actually ones that are generated as Gaussian distributions of our red cell and platelet histograms. And they are equally important in our interpretation of abnormal scattergrams. One thing we will notice is that pre and post the Gaussian peak, that they should return to normal. And that when they don't, this is our first evidence that perhaps we have a pathologic condition and perhaps we should investigate further. We'll also find that the Sysmex allows us to do, or actually provides us statistics. So it provides us what we call a red cell distribution width, and it provides us an SD or a CV of these, and the larger our statistics, the larger the distribution. So again, it becomes a trigger for us to look at what could be causing a greater variation in either red cell or platelet size. All right, so let's look at some differences between some various species. So this shows some normal WDF scattergrams for cat, dog, and horse. I particularly called out EOs because I thought this was actually an interesting one. So on the left, we have a cat sample. This is drawn, labeled as 0 hours, meaning this is a fresh sample. The dog as a fresh sample, the horse a fresh sample. Next to it, we now have a cat, a dog, and a horse. It's the same sample, but now at 72 hours. So the important message of here, if you look at anything at 72 hours, you lose the distinct separation of population. Most important, whether a human or a veterinary sample, is actually the appropriateness of your sample. So the age of your sample and the viability of your cells. Once you pass that, as I've commented before, not all cells, not all species are going to look absolutely identical. If you look at the cat and the dog, we have kind of this linear distribution of neutrophils, lymphocytes, monocytes. Both of them kind of have that. If you look at the horse has a greater degree, typically, that the neutrophil population is pushed farther on the x-axis, more typical to what you would find in humans. And I will tell you, monkeys kind of do this same application. So the importance of this is that they can be different. One important application I saw in this publication was actually where eosinophils fell for this particular cat sample. And you can see they're actually very high on the y-axis, where typically, and in most of our examples, we expect them to be right next to the neutrophil population. So indeed, they can vary. And I've given you examples of various eosinophils that occur across species. And you can see that although they are very similar, they are not the same. For instance, if I look at the equine or the dog, I actually have these very, very large globules. Their granulation is extremely large. In contrast, the granulation of cat eosinophils is usually very small, very compact. Because we are measuring light scatter, these small differences can make large differences in where the population actually resides. All right, so let's look at where some of these abnormal cells reside within our WDF. And I've included two different diagrams for whichever one you find easier to interpret. One that looks more like the scattergram on the right, and one that maybe looks a little more like a cartoon on the left. But whichever serves you better to learn this, please go ahead and use that. All right, so we've talked of four population. Here's our neutrophil and basos in our aquamarine on this right-hand diagram. Here's our lymphocytes in pink, our monocytes in green, and our eosinophils to the right and in red. That's typical. As I said, some may be slightly different. The populations we see most frequently, one of the most frequently is a left shift or IG, immature granulocytes, bands, metamyelocytes, myelocytes. These cells are larger than normal mature neutrophils, so you would expect them to be in the same position as neutrophils, but higher on the y-axis, because this is going to mean I have a larger cell, I have a larger nucleus, I should have greater fluorescence. So they will be right above your neutrophil population. Another population we haven't talked about is our nucleated reds. We said we like to assess them from the WNR, but you can likely still see that they're going to be present on the fluorescent scattergram. Typically, they are the size of a red cell, but they have a nucleus in them. So we would expect them to be smaller than our lymphocytes and typically below them. So NRBCs, nucleated red blood cells, can be seen, even if they're enumerated on the other. Larger cells, atypical lymphocytes, blast cells, all of them have much larger nucleus, much higher nuclear to cytoplasmic ratios. This high nucleus, this high DNA content will cause them to have very high fluorescence levels, and we would expect them to be much higher on our y-axis. So with that, we'll start our case studies. And our first one was gathered from the XT analyzer, and it's an example of immature granulocytes. We can see it indicated on our scattergram in dark blue and indicated with an arrow of IG, immature granulocytes. When you're interpreting scattergrams, you should always look for where are your normal populations. We should have a normal debris. We do. We should have normal lymphocytes. We do. We should have normal monocytes. We do. We should have normal neutrophils, normal eosinophils. We do. But in this case, we actually have something extra. We have this population that the analyzers recognizes as different, the neutrophil population. And it actually flags it for us so that when we look at our vast number of parameters that come, we can say, do I have a flag? The answer is yes, I have a flag. I think we are dealing with immature granulocytes. And in this case, it does indeed flag for us that I have high numbers of IGs, both percentage and absolute, and it triggers us then to do a microscopic differential. So the abnormal scattergram becomes that first step to say, I need to go further on this one. And with this one, with the manual diff, we can see we have a slight elevation in bands and actually a few metamyelocytes. So it was an accurate predictor that immature granulocytes were present. I use this one, and as I said, I would use a few human applications, and that's because some of the flagging has been enhanced with the XN application. And this is actually a very nice feature to have. In this particular application, human albeit, again, we do the same thing. Where are my normal populations? So I have lymphs, I have monos, I have neutrophils, I have eos. Again, in this one, I have an even larger population of immature granulocytes, so that you would expect as you trigger a manual microscopic differential, you would see more immature granulocytes. But beyond that, with new flagging, we find that the XN is actually going to tell us where on this x-axis the midpoint of this position really resides. And this gives us an estimation of whether these neutrophils could be hypogranulated if they're shifted to the left, or hypergranulated if they're shifted to the right. And why is that important? It becomes, again, another piece of the puzzle and another important indicator. In this case, you're looking at pocket granulation. It increases the granulation of the cell. You have hypergranulated cells, and is consistent that this individual may have an infectious process. All right, so next study, and I purposely didn't label this one so that we could go forward through it and see as this particular animal progresses. And it's a dog, and here's our first day. We do the same as we always do, lymphocytes, monocytes, neutrophils, and eos in their normal population. And we can see with the circle that there is a small population above the neutrophils. So we might suggest that on day one, when this dog was seen, that there might have been a low level of immature granulocytes. As we progress to day 5, even without looking at the values that are obtained, if you have day 1 scattergram and day 5 scattergram, you can say this animal is different. And this animal is different in that they have greatly increased their number of immature granulocytes. Beyond that, we actually have a population, a small population that's even higher. And that might question us of whether this animal has such a pronounced left shift that there might be presence of blasts. Again, you're only going to really resolve this through a microscopic differential, but it is consistent with the actual diagnosis of this animal, which is actually sepsis. All right, next one, again, an ASCP example. And this one I put because I think it has a nice application of what you can do with the Sysmex. All right, so this one was on an XT, and this was a dog again, and seemingly pink lymphocytes, green monocytes, and I now have 2 gray populations. Well, what happens when the analyzer is uncertain of what that population is? Instead of giving it the color that you're looking for a specific cell type, it may gray out the population. If you were looking at this and say, well, if it's a normal dog, this will be the one on the left is the neutrophil, and the one on the right is going to be the eosinophil module. And as this person did, they reanalyzed it. Okay, so one of the features of Sysmex is I can reanalyze these and I can actually move the gates. And they did so. They made this my EO population, seemingly EO population, neutrophil. And you can see they actually even moved the lymphocyte population. And if I had to pick it, I probably would have picked where they picked as well. So we now have one that looks more like a normal population, except that this EO population looks really higher than normal. It's a really big, robust population. So you would never want to accept a reanalysis without looking microscopically. But it is, again, a good indication that something needs to be done. And that's something. In this particular case, it was a canine eosinophilia. You certainly would look at your data, and your data tells you that it had high EOs. But again, confirmation comes down to a microscopic differential so that you can confirm what is there. And if indeed you confirm, there's no reason that you cannot report out your reanalysis values, but you do need that confirmation. All right, case 4. All right, so case 4, again, a dog, and it looks kind of similar to what we've been looking at in our previous studies. But in this particular case, we see that instead of this entire population being stained green, I actually seemingly have these cells that are wrapping around the sides of the mono, and the ones on the top are actually a different color. This leads me to question that these are truly monocytes. So certainly drives you to looking at your data more closely. And in this case, it says, okay, I do have somewhat higher lymphocytes, but I don't really have any other clue of what might be going on. So you, in this case, would also want to be able to do morphology. And in this particular case, it would not be uncommon that you might find cells that look like these. So these indeed are atypical lymphocytes. Some individuals still call them viral lymphocytes. And they are large lymphocytes that have abnormal staining. The blue staining sometimes is consistent with the production of immunoglobulins by these lymphocytes. And because they are larger, they fall in a category where they would have a larger nucleus, and they would have greater fluorescence, and they're different from our normal lymphocyte population. And this indeed was a case where of viral infection and or reactive lymphocytosis in this particular dog. All right, so another change in the XN as far as flagging, and I put this here because some of the flagging differences is kind of nice too. So it uses, again, gating of cells that are larger than our normal lymphocyte population, and uses an algorithm to detect whether there's a difference. And we will get flagging to let us know that we potentially have reactive lymphocytes or potentially have activated monocytes. And those that are actually high enough on the y-axis potentially could be plasma cells or other types of blasts. All right, so I've included for monocytosis a couple of human ones since I didn't have the veterinary counterpart for the XN, but I thought they were still a good learning opportunity. So looking at this particular scattergram, we again find lymphocytes in purple, monocytes in green, our neutrophils are blue, and a very, very scant eosinophil population. They all look pretty much in a normal population. This one, the monocyte population might be a bit higher on the y-axis, but it's pretty much in a normal position. Therefore, we might expect, okay, because of the magnitude of this, we would hope that when we look at the values that we would have higher numbers of monocytes, such as in a single field, but their morphology might actually be quite normal, as in this case. In contrast, and again, this is an XN scattergram, and this one came with a diagnosis of acute monocytic leukemia. And you can ask yourself, was this different from the last one where it was just a monocytosis? And the answer becomes clearly yes. In this case, we have probably a set of monos that are down here in a normal size and maybe normal morphology, but we have a pretty large population of monos that are shifted, or in mono area, that are shifted fairly high. So that when we do this review microscopically, more characteristically, you are now going to find blasts. And in this case, they were characterized as monoblast. They are large, they have monocytic characteristics, very fine blue cytoplasm, extremely large nucleus, very prominent nucleoli, and again, a case where the scattergram leads you to looking for an acute monocytic leukemia. So you may ask, well, does the XT or the XN, could you be aware of a leukemia in an animal since you've just shown me one that occurs in people? And that answer is, again, yes. This particular diagram is, again, taken from a dog. It certainly looks different from the normal scattergrams that we've looked at. Basically, the only thing that looks normal is perhaps the debris in dark blue and the eosinophils in red. At this point, it's like, okay, these are likely my neutrophils and perhaps some immature granulocytes, but the analyzer can't tell what this is, so it's graded out. And you're going to guess it's in the area of lymphocytes and monocytes, so it's likely lymphocytes or monocytes, or both. And it is pretty high up on this scale, so we question whether there might be blasts there as well. So here it is as far as numbers with it. And indeed, a very high white count, typical of leukemias. Okay, so a 69,000 white count. We have small numbers of immature grands, so what we're seeing going upward here might be immature grands, but the very most important thing is that it's actually plussed out or minused minus the lymph and mono population. So telling you in this particular case, you absolutely need the microscopic differential. But the sample did indeed give a blast flag, so it leads us in that direction. And here we are. So in this case, again, we have high numbers of cells, and we have cells, in this case, in this differential, they saw 7% monoblasts, so fairly small numbers, but significant numbers of monoblast. But 49% monocytes, in which this person who did the differentiation Differential actually classified a lot of them as pro-monocytes, which attributes to the size. Lymphocytes were actually quite normal. And we can see that the classification for this, in this particular dog, was an acute myelomonocytic leukemia. So indeed, the XT and XN analyzer can predict for you the presence of leukemia in animals. All right, so let's look now at some of these RBC and platelet Gaussian curves, because these two can be considered scattergrams, and they also tell us whether we potentially have abnormalities. When we look at this RBC scattergram, we have a pretty good Gaussian peak, but we also have two other things. So we have this peak that occurs before the threshold, and we have a minor peak that occurs after. Remember, these indicate that red cells are larger as we go to the right on the x-axis, and that red cells are smaller as we go left on the x-axis. Small bumps on the x-axis on the right side often refer to small bumps on the left side typically refer to small red cells, so they may indicate red cell fragments. Important to look, as always, at the values that the Sysmex actually gives you. And with this one, we can see we have an animal that has a pretty profound anemia, one with a 7.3 hemoglobin. I talked about the analyzer being able to give you statistics for your RDW and as both an SD and CV. And we can see in both cases they are highlighted. It means that these extra things we are seeing contributes to the broader SD and CV, telling us that there is a difference in the red cell size present in this sample. We do have some level of retics, 5%, okay, so a small number. And we're also flagged on this sample that there are nucleated reds. And we're also flagged that I have a difference between my impedance and my optical platelets. So I have multiple things to look at. I have to look at both of these. What is the cause for these two extra bumps on both sides? And do indeed I have nucleated reds? So we look at the other scattergrams from this particular animal. Here is the retic, okay, and with the retic, I can see, yes, I do have an increased number of retics indicated by this pink and red in my reticulocyte area. But the most important thing from this scattergram is that the red cell themselves are actually shifted downward, so that these cells in our red cell population are actually erroneously elevating our platelet count. As I said, this was a reason that we went to using a Platelet-O. We can see there is greater separation between our red cell population, blue on the top, and our platelet population, which is on the bottom. Both of them, retics and platelets, in this particular case, show us that we have increased production of both red cells and platelets. When we looked at the WNR, we want to see that we have nucleated reds. And indeed, we have an extra population that is consistent with the nucleated reds that were reported on the analyzer. And again, our final confirmation, as always, goes to microscopic evaluation. And in this one, you're looking for red cell morphology. So you're looking for something that correlates to that bump on the left saying, do I have small red cells? And indeed, I have multiple small red cells in here, probably enough to make a visible bump that we saw on the scattergram. Do I have nucleated red cells? Certainly, I have nucleated red cells that are there, consistent with increased reticulocytes. All right, so let's look at another one, and I've labeled this one for you as hemoglobin turbidity and lipemia. The important thing is not every bump to the left is a small red cell. And one of the common ones that you can get in practice is increased hemoglobin turbidity or lipemia. So when you look at this set of data, you're going to, again, look at the things that flag for you. And one of the most prominent ones for these are your indices, MCH, MCHC, or mean cell hemoglobin concentration. In this case, I have nearly a 50 for my mean cell hemoglobin concentration. Hemoglobin is a protein, and as such, it has a size to it. And as such, only so much hemoglobin protein can fit within the size of a red cell. Consequently, hemoglobin analyzers typically pick a value, and with that value, you know the maximum amount of hemoglobin that can fit into a red cell. For most analyzers, this is about 37 grams per deciliter of hemoglobin. When you exceed those values, what is causing the color change in your hemoglobin determination, remember that is a spectrophotometric read, is something that is adding extra color. So it is either extra free hemoglobin, it is extra turbidity, or very commonly lipemia. So these high values are very important in your diagnostic evaluation of this scattergram. Also important for you to look and check whether the hemoglobin and hematocrit match. If we remember the match, hemoglobin should be approximately, if you multiply it by 3, should equal your hematocrit. We can see if we multiply this one, 11 by 3, it would give us a hematocrit of about 33. So we have a mismatch between hemoglobin and hematocrit. Fortunately, with the analyzer, it does indeed flag turbidity, hemoglobin interference. Whenever MCHC is greater than 36.5, it is then up to us to evaluate what that reason is. Is it lipemia? Is it hemolysis? Is it Icarus? What is falsely elevating the hemoglobin? Some labs will also do a spun hematocrit to confirm that the hematocrit is correct. And all labs would likely do a plasma replacement to try to get rid of the interfering color so they can have an accurate hemoglobin reported and then recalculate their indices so they too are accurate. Looking at platelet histogram. Okay, we're going to move to a couple case studies with platelets. Just as the red cells, we would hope that it returns to 0 on both ends of the baseline so that there is lack of interference in this read. All right, so here are 2 different dog histograms, again, from an ASCVP presentation. The one on the left is a normal dog. We see good separation of red cell and platelets, okay, and we see pretty good size-wise population. This is a normal dog. The one on the right, we see, again, a pretty good population of red cells. We see a much higher level of reticulocytes than our normal animal. But the big difference we see now is a lack of platelets. We have a very, very small platelet region, and we find that this population of immature platelet fraction is the response to the lack of platelets. All right, so I included 2 more platelet case studies, and I included human ones because the new XN platform has that new channel, that Platelet-F channel, and it's extremely important for accurate platelet counts to include and to consider the results that are seen with a Platelet-F channel. I thought this was an excellent example, albeit human, taken from the University College of London, and they used beta thalassemia major patient. And we know in that patient population, there's going to be numerous fragmented cells, so you will have a lot of small red cells. When you look at that platelet impedance count, you're going to find that there is no baseline between the platelet bump and the red cell bump. It should not be, you should recognize, even looking at this one, that the platelet impedance count is giving you an erroneously high count due to these platelet fragments. But when they ran them, they looked actually at the XE platform and they looked at Platelet-O. We said, well, that should be better. And indeed, it looks somewhat better, but you can see that there's still an overlap between these very small red cells in the dark blue and our platelet population in that aquamarine. And if you can see the presentation there, we can see platelet I was about 500,000, whether it was measured on the XE or the XN. So the platelet I told us this patient had 500,000 platelets. When we looked at Platelet-O, it was 111,000. And when we looked at Platelet-F, which in this case uses a fluorescent probe specific for platelets, we saw 140. So this tells us, in this particular case, there was about an 80% error if you had used a platelet I. They confirmed this by actually running on flow cytometer as CD61, which is a platelet marker, and they showed that indeed the count was 152. This shows the value of actually having a fluorescence-based count and the value of the Platelet-O versus the platelet I when there are numerous small fragments. And I included this one, okay? This is another example of using the Platelet-F Scattergram. This is in a case of acute pro-lymphocytic leukemia with chemo and the presence now of white blood cell fragments. Although this is human, certainly we have multiple diagnostic areas where animals are receiving chemotherapy, so it's still a very relevant example. So let's look on the left. On the left, we have our XE and our Platelet-O, and we're saying that many times Platelet-O can be better than the platelet I. Important not to make that a hard and fast rule. It's important to look at the scattergram and determine what that scattergram is telling you. When we look at this Platelet-O, we ask ourselves, does it look normal? And certainly it does not. Actually, our red cell looks pretty small. We do have some retics. It's not too bad. Our problem comes when we start looking at this platelet, and we actually have a level of platelets on the bottom. And then we have this huge mass up here, which the analyzer has tried to divide between what's the immature platelet fraction and what might be best. And by doing it, it gives us a total platelet count of 181,000 for this particular patient with leukemia. Same patient run on the XN platform with the Platelet-F. Now when we look at the Platelet-F, okay, we have really a good separation between red cell and platelet. And our platelet parameters seems pretty consistent, okay? We have a nice population that seems pretty consistent, not one that seems, okay, what's this stuff down here versus what's the stuff that's above it. So let's count. The platelet I and the XE was actually 25,000. A level that would likely be in a critical range for a leukemic patient. The platelet I was 28,000, so a good correlation between platelet I, good impedance counts for both of them. Here's our Platelet-O, and this is why I said not always better, was 181,000 because of this big mass that was here. And now we compare it to the Platelet-F, and the Platelet-F is about 24,000. Huge difference and very significant diagnostic difference that you would be treating, in this case, a patient, and assuming they have a very normal platelet count when indeed they have a very reduced platelet count. Beyond that, the platelet fraction, the immature platelet fraction in the XE value gave a value of 41%. So the physician might think the bone marrow is actively trying to compensate and make platelets. Whereas in the Platelet-F, we have a 1.1% immature platelet fraction, showing us that we have very little new release of platelets. So I'm really quite excited about this parameter, and I think for our leukemic and chemo populations, it's going to be a nice addition to the repertoire. All right, we're winding down here. So one more on RBC. Okay, we have another little bump on the left. Okay, so you're saying, okay, well, it's small red cells or it's turbidity. And the answer is, well, maybe not. Okay, in this case, again, we have a platelet that's flagged for us. We have a high MPV on this particular one. You know, platelet I and platelet O, they didn't have a flag between the two of them. But when we look at the platelet histogram on this one, we see that it doesn't quite come down to baseline. And in essence, this actually was a rat sample, so the 700,000 platelets was actually kind of low for this particular species. And we can see that they are forcing new generation of platelets, which are these giant platelets that are there. When we look at now our Platelet-O, we can see that exactly that's what this rat is doing. We have pretty normal populations, but they have a pretty big immature platelet fraction. So it works pretty nicely in animals, especially for seeing regeneration when you want to see regeneration. All right, one more platelet in animals. And again, this kind of looked like the thalassemia example that we had from the London Hospital in people. So you can see we get these in animals too, where the platelet just doesn't go down to baseline and the red cell continues up from it. When we look at this one, it now tells us, okay, I do have a platelet flag, and I'm not really pleased with my platelet I. I'm also flagged for that. But other than that, this animal looks pretty fine. They don't have an anemia. Everything else looks fairly decent, except we have some high statistics because these scattergram is abnormal. All right, so it's a very common problem. This one was actually platelet clumping in a dog, and we know that in animal samples, because of the difficulty in drawing, it can be very common to get some interference from platelet clumping. So could this Platelet-O tell us that there might have been something there? With them, we can see that we kind of have now, instead of a discrete population of platelets to the left and that nice immature platelet fraction, we now kind of see a smear of platelets. And that's because we are seeing now platelets being assessed as clumps of fluorescence. So whenever you see this clumping, this smearing, it's synonymous with truly a clumping that you would see morphologically. Okay. All right, another one that was given by ASVCP and I thought was relevant for this population and a good learning example was looking at the reticulocyte scattergram and where those population falls. Typically, normally, you could draw a line through the midpoint of your red cell population and your retics will fall right on it. And your red cell and your platelet populations will have a good separation. This particular example showed us microcytic red cells with iron deficiency in a dog. We can see when we draw that same line down the middle that the new cells that are being produced are actually pretty small because of the lack of iron to fully make those hemoglobin molecules. So this they labeled as a microcytic iron deficiency anemia. And you can ask yourself, do I have problems with any of my other counts? And you might say, if I could have a problem with a platelet count. And this you might want to evaluate simply because those new cells are actually so small in this animal. So again, an enhanced flagging. The XN will actually do a flagging, and it will do that measurement for you. How far is the red cell from my platelet population? And how far, and can I determine whether I am seeing a level that might be synonymous with hypochromasia or not? So these additional flaggings help us in really determining microcytic hyperchromic anemia, whether they're in people, as this demonstrates, or animals. It just is going to measure and flag. And here is that iron deficiency now in a dog, showing you that it actually exists too. We can see in those cases, I'm not seeing a bump, but I'm seeing that my Gaussian curve actually goes over the threshold because the cells are smaller than normal. They fall below the threshold. And when I confirm microscopically, I do indeed see those small cells. I do see hyperchromic cells. And just as we've looked at a couple times, I see that the platelet count fails to return to baseline. And I will likely have to look at my platelet count to make sure I'm reporting an accurate level. The last two, okay, the very last two we look at now are a case of parasites, and certainly we have parasites in animals. This one, again, happens to be a human, and this one is the only one that's actually taken from the XE. But I take it because it is relevant. You see parasites in animals, and you can see them pretty consistently, so it's an important learning lesson. The two cases on the left were humans that had no malarial. They were malaria negative. You can see normal populations within their WDF and normal populations within their WNR. When three other individuals who had Plasmodium vivax were analyzed, you're not going to see a signature, but you see differences in all of them. If you look closely, you can now see I have multiple populations. Where you should have a single neutrophil, I now have two aqua, or I have two gray categories. I also look like my EOs have multiple population. In my WNR channel, I have, although difficult to see here because it's dark blue, but I have some arrows, I have more debris, and the debris is shifted off to the left. So why do I have more debris? And why do I have more populations? And in this case, indeed, it was malaria. Okay, so malaria is one, particularly if you're working in monkey colonies, and you know that we have malaria there. But you could have babesiosis, you could have a number of different parasites. It's important to remember that these parasites can have very large nuclei in them. They indeed can stain with this DNA stain. So they can be seen as another separate population. And also these parasitic infected cells sometimes are kind of lice resistant. And because of that, we see larger debris populations. And the very last one, okay, also related to our debris population, but back into an animal. This is actually a sickle cell model showing that lice resistance. So with it, you can see that I have a debris population, but just like platelet clumps, I have this big smear of something. So it shows that there's a problem in this particular sample, and in this particular one happened to be a sickle cell mouse model showing a level of lice resistance. So to summarize, important to know that the knowledge of seismic scattergram interpretation is a very useful first step in hematologic disease diagnosis, both in humans and in your veterinary species. And when we learn and understand the pathologic changes in these scattergrams, it's going to be similar whether we're using an XE, an XT, or an XN platform. So then you might ask, well, what does the XN platform give us that the XT doesn't? And really, there's a number of things. It improves throughput. We can actually combine two analyzers together. So really, our efficiency and throughput can be improved. It decreases the amount of sample volume that we need. And for small animals, particularly maybe like mice, that's an important criteria. I really like the improved flagging of our abnormal scattergrams, so it helps you not miss these abnormal findings. You have an automated reporting of nucleated red blood cells, that access to the Platelet-F channel that we talked about to further minimize erroneous platelet counts. And lastly, what we didn't speak about was actually the automation of a 7-part diff that can be used for body fluids that's now available on the veterinary platform. So thank you.
And thank you very much, Dr. Criswell, for that informative presentation. We will now start the live Q&A portion of our webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question drop-down box located on the far left of your presentation window. Type your questions into the box that appear on the screen and click the send button. We'll answer as many questions as we have time for at the end. So let's take a look at our first set of questions coming in from our audience members. And thank you again for your participation. Dr. Criswell, why is multi-species software necessary to accurate WBC differential discrimination?
Okay, so I think we covered that in part of the presentation, but let me hit on it once again because it really is an important aspect. You know, I will tell you 20 years ago, people ran all their veterinary samples through the human-based hematology analyzers. And it took a while to recognize that they really aren't very accurate. So it's absolutely essential to use a veterinary software when you're reporting veterinary results. We pointed out that although they're morphologically similar, white cells vary between species. So they can have different sizes, they can have different shapes, they can have different granularity. And these subtle differences allow the cluster designations to vary sometimes substantially. So because of that, having an accurate differential is important. Equally important for having accurate cell counts, because the sizes of platelets, the sizes of red cells vary largely between species. So it's absolutely essential for accurate counts and accurate differentials.
Thank you, Dr. Criswell. Our next question is, what is the benefit of having the ability to set your own WBC differential gating?
All right. So we saw one example where they actually reanalyzed a sample. So that's one advantage, that if you can really see how it's separating, you can go through a reanalysis and still obtain your data. Beyond that, we've actually found that you can use it for maybe even alternate applications. We've gone through and actually created a differential performed on bone marrow. And you do that by knowing what's in those populations and by defining those populations. So that can be an important thing. You know, if you're doing a research study, I can say it's also important that you could set your gate. And then you always know that it's exactly where it's going to be. And the last example I should give in the veterinary world is you may not know the species you're going to deal with today versus the species you deal with tomorrow. So being able to set up your own, you can go past the normal veterinary species that are provided, and you can really analyze anything. And the example I can give is in my past life, we actually supported an aquarium. So the question came, well, can you do a CBC on a shark? Can you do a CBC on a whale? How about a sea lion? And the answer became, yes, we can do it on all of them. And were they the same? No, they weren't. But it allows you flexibility, which you really need in a veterinary world.
Thank you, Dr. Criswell. Our next question, is the Sysmex analyzer capable of performing differentials on samples other than peripheral blood?
Yeah, so I just mentioned it briefly in the summary. Certainly the XN now allows body fluid analysis, which is a nice new thing. And with the ability to set channels, I would say even on the XT, some of us would do things that way, although certainly better to have it, that it's set up and actually fully validated so that you aren't doing the validation process yourself. But the answer is yes. And I also alluded to we've been able to do bone marrow differentials. So the answer is yes, you can use it for other fluid types.
Thank you so much. And I want to thank our audience again for their questions, and any questions that aren't answered today will be receiving emails to your questions after the presentation. Dr. Criswell, would you like to provide the audience members with any closing remarks before we close today?
Well, I guess I will say I've had a long history of looking at hematology scattergrams. I've also always found them fun and informative. I hope the audience have enjoyed it. And I would encourage them to learn more about it, because it's going to provide better service to your customers by knowing what you're looking at in these abnormal scattergrams.
Wonderful, thank you. And thank you for your presentation and for your tremendously important research. I'd also like to thank LabRoots and Sysmex for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You'll receive an email from LabRoots letting you know when this webcast will be ready for a replay. Please share this announcement with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us. We hope to see you again soon. Bye-bye.